The previous session, we have completed the first part of the component, that is, cells of the connective tissue. To recap, it was discussed on introduction, functions, composition, and 8 types of connective tissue cells. In this session, I will be discussing on extracellular matrix. Intercellular or extracellular matrix is a non-living material synthesized by connective tissue cells, particularly fibroblasts. It consists of fibrous and non-fibrous element. The fibrous element has 3 types of fibers: collagen, elastin, and reticulin. The non-fibrous element is formed by the ground substance. Ground substance or amorphous substance is highly hydrated, viscous, semi-fluid, and transparent gel-like substance. It is composed of complex mixture of macromolecules like water, electrolytes, glycosaminoglycons, proteoglycons, and multi-adhesive glycoproteins. It fills the space between the cells and fibers. In other words, cells and fibers of the connective tissue, as you see in this picture, are embedded in the ground substance, like that of ice cream embedded with fruits and nuts. Imagine ice cream as ground substance, fruits and nuts as cells and fibers. The functions of ground substance are: it provides morphology and framework for the tissue, it protects and binds connective tissue cells together, it acts as a lubricant. Through diffusion, it acts as a medium for the exchange of materials like nutrients and metabolites between the cells of the connective tissue and blood capillaries. It also prevents the spread of microorganisms. When bacteria is spread in ground substance, they release spreading factor called hyaluronidase, which produces depolymerization of hyaluronic acid and removes the chief obstacle to the spread. It acts as a mechanical barrier for free movements of particles or other dissolved matter in tissue space and to the penetration of invaders. It helps in storage of water, which is provided by the large surface area of carbohydrate molecules of hyaluronic acid. With advancement of age, amorphous element of matrix diminishes and fibrous element increases. Glycosaminoglycons, also called as GAGs, in the ground substance are long-chained unbranched mucopolysaccharides. They are negatively charged, attract osmotically active cations such as sodium ions, and make the matrix hydrated gel through which water, oxygen, and solutes pass through to reach the cells. Based on their chemical structure, 2 types of glycosaminoglycons are present: non-sulfated and sulfated groups. Non-sulfated type includes hyaluronic acid. Hyaluronic acid holds much water or tissue fluid because it possesses long chains. Hyaluronic acid keeps the ground substance in salts. Hyaluronic acid is abundantly present in the skin, where it makes the tissue soft and pliable. Sulfated type includes chondroitin sulfuric acid, dermatin sulfate, heparin sulfate, and keratin sulfate. Chondroitin sulfuric acid presents 3 principal types: chondroitin sulfate, A, B, and C. They impart cohesiveness to tissue and keeps the ground substance in the form of gels. It is abundant in cartilage. Proteoglycons are large molecules consisting of a core protein to which various numbers and combinations of sulfated gags are attached. However, amongst the different glycosaminoglycons, hyaluronic acid, which is a non-sulfated group, does not bind to the core protein and hence it does not form proteoglycan. Core protein of proteoglycan binds with long strand of hyaluronic acid through link proteins. Example is agrican. Glycoproteins are proteins to which oligosaccharide chains are attached. They bring about adhesion between cells and extracellular matrix. There are several types of glycoproteins in the ground substance. Fibronectin, it is a multifunctional glycoprotein. It mediates the adhesion of cells to the extracellular matrix. Laminin, it mediates the adhesion of epithelial cells to basement membrane. Entactin, it is a link protein which binds laminin to type 4 collagen of basement membrane. Osteonectin, it is present in the bones and plays an important role in bone mineralization. Chondronectin, it is present in cartilages. It mediates the adhesion of chondrocytes to the extracellular matrix. To look at the second component of extracellular matrix, fibers. Fibers are part of the ground substance of connective tissue. They are elongated structures formed from proteins. The proteins of the intercellular substance are polymerized after secretion by fibroblasts and organized to form fibers. There are 3 types of fibers: collagen, elastic, and reticular fibers. Collagen and reticular fibers are composed of protein collagen, and elastic fibers are composed of protein elastin. The functions of fibers are: they provide strength to the connective tissue. They are distributed unequally among the different types of connective tissue with predominant fiber type which are responsible for conferring specific tissue properties. Let us see the details of each of the fibers now. To start with collagen fibers, this is the spread of loose areolar tissue in which bundles of collagen fibers are observed. The various fibers, sheets and networks made of collagen are all extremely strong and resistant to normal shearing and tearing forces. It is the key element of all connective tissue, epithelial basement membranes, external lamina of muscle and nerve cells. It is most abundant in body, representing 30% of dry weight. Characteristic features of an individual collagen fiber is it does not branch and is thick and wavy. It is made of protein subunits called tropocollagen. Collagen fibers are eosinophilic in nature, that is, it takes up pink or eosin stain. Collagen fibers are distinguished by their morphologic characteristics, molecular compositions, distribution, function, and pathologies. There are around 28 types of collagen which are grouped into several categories. The most common forms are type 1 present in tendon, ligaments, dermis, fascia. Type 2 collagen is present in hyaline cartilage. Type 3 collagen is present in reticular fibers. Type 4 collagen is present in basement membrane. They provide toughness and tensile strength to the tissue. Coming to the second type of fiber, that is elastic fibers, they are thinner than type 1 collagen and is interspersed among collagen bundles. Elastic fibers have the property to stretch in response to tension and hence provide elasticity to the tissue. Individual fibers are thin, branch, and form three-dimensional network. They consist of protein elastin and microfibrils formed of protein subunits, the tropoelastin. Elastic fibers are eosinophilic. As mentioned earlier, collagen fibers are also eosinophilic. Hence, in H&E, that is amidoxylin and eosin stained preparations, it is difficult to distinguish between these two fibers. Since collagen fibers are in bundles and elastic fibers are in single, if you take little care and observe, you can identify both. Elastic fibers help recoil and restore the normal shape of the tissue after distortion or stretching. This is because a network of elastin molecules is formed by covalent bonding, which can stretch and recoil. The tissues that have high concentration of elastic fibers appear yellow in color on naked eye appearance. These fibers are present in large numbers in skin, lung, large blood vessels, elastic ligaments like ligamentum nuchae. In large blood vessels, it is present in the form of elastic lamellae. They stain poorly in H&E. As the age advances, the elasticity is reduced. Coming to the third type of fibers, reticular fibers, they are thin, delicate fibers which are arranged in extensive network. Reticular fiber consists of type 3 collagen. They are thinner than other types of collagen fibers. They are not stained by H&E stain. They are stained black by special staining called silver impregnation method, and the fibers are termed argyrophilic fibers. These fibers form the framework in lymph nodes, spleen, bone marrow, liver, basement membrane to provide support to the cells in these tissues. They surround adipocytes, smooth muscle, nerve fibers, and small blood vessels. Also serve as supportive network stroma around parenchymal secretory cells and rich microvasculature of endocrine glands. Reticular fibers are produced by fibroblasts. With this, we have completed the second part of connective tissue, that is the extracellular matrix. In the next session, I will be discussing on classification of connective tissue.